For decades, the answer to the question of what medicine could do for osteoarthritis cartilage was honest and disappointing. Not much. We could reduce the pain. We could reduce the inflammation. We could slow the rate of damage with the right lifestyle and exercise and dietary choices. But the cartilage itself, once it thinned, once it degraded, once the smooth cushioning surface that separated bone from bone had worn down, medicine had no way to grow it back. That answer may be changing. In November of 2025, researchers at Stanford Medicine published a study in the journal Science that has generated more scientific excitement in the osteoarthritis field than almost any finding in the past decade. Their study found that blocking a single aging-related protein, a protein called 1,5-PGDH, caused damaged knee cartilage to regenerate in older mice, producing results that the study's own authors described as dramatically greater than anything previously reported with any other drug or intervention. And this was not the fibrocartilage, the inferior scar-type cartilage, that sometimes partially fills cartilage defects, but hyaline cartilage, the smooth, load-bearing articular cartilage that osteoarthritis destroys, and that medicine has never been able to rebuild. Simultaneously, in 2026, a stem cell therapy called CARTISTEM, already approved in South Korea and used in over 36,000 patients, received FDA clearance to begin its pivotal phase 3 clinical trial in the United States and Canada, aiming to become the first stem cell therapy approved for knee osteoarthritis in the American market. And a comprehensive review published in the European Journal of Pharmaceutical Sciences in February of 2026 catalogued the current state of the entire field of intra-articular disease-modifying osteoarthritis drugs, the compounds specifically designed to restore cartilage rather than simply manage its destruction. We are at a genuine inflection point in the science of cartilage treatment. Not a moment of arrival. The therapies that could transform osteoarthritis care are not yet in your physician's office, and some may not arrive for years. But a moment of credible, evidence-based hope that deserves honest, specific, scientific explanation so that the millions of adults living with osteoarthritis can understand what is actually happening in the research, what it might mean for their future care, and what the distinction is between genuine scientific progress and the health supplement marketing that uses the word breakthrough without any of the evidence that justifies it. Today I am going to tell you exactly what the science says about 3 specific categories of injection-based cartilage treatments, what they are, what they do at the biological level, what the human evidence shows, and what the realistic timeline is for each to reach clinical practice. Not marketing, not hope disguised as fact. The actual science, explained clearly. Before we begin, subscribe to this channel right now if you want evidence-based health content that follows the real science of arthritis and joint treatment. We create this specifically for adults who want the truth about what is happening in research and what it means for them. Hit subscribe. Let us start with what makes cartilage regeneration so difficult, because that context is what makes the 2025 and 2026 breakthroughs so significant. Articular cartilage, the hyaline cartilage that covers the ends of bones in synovial joints, is one of the most metabolically inert and least regenerative tissues in the adult human body. Chondrocytes, the cells that produce and maintain the cartilage matrix, are terminally differentiated cells that lack the active stem cell populations present in most other tissues. They exist in an avascular, unrural environment with no blood supply and no nerve supply, and they receive their nutrients and remove their waste entirely through the slow diffusion of synovial fluid through the cartilage matrix. This metabolic isolation, combined with the mechanical compression loads that cartilage endures with every step, makes cartilage profoundly resistant to the repair processes that most tissues activate after damage. When cartilage is damaged, whether by mechanical injury, by the inflammatory environment of osteoarthritis, or by the age-related changes in chondrocyte function that accumulate over decades, the chondrocytes attempt repair, but cannot match the rate of degradation. The matrix metalloproteinases that destroy the collagen and aggrecan of the cartilage matrix, activated by the same inflammatory cytokines we have discussed throughout this arthritis series, outpace the chondrocyte's capacity to rebuild what is being degraded. The result is progressive cartilage thinning, exposed subchondral bone, and the bone-on-bone contact that produces end-stage osteoarthritis. The central challenge of cartilage regeneration medicine has always been the same. How do you transform a tissue that has lost its capacity for self-repair into one that can rebuild its own structure? The Stanford 1,5-PGDH discovery represents a different answer, and it is the reason the scientific community has responded with such unusual excitement. The protein at the center of the Stanford research is called 1,5-hydroxyprostaglandin dehydrogenase 1,5-PGDH. It is classified as a gerozyme, a protein whose levels increase progressively with age and whose activity contributes to the functional decline of aging tissues. The Stanford team, led by Drs. Helen Blau and Nidhi Bhutani, had previously identified 1,5-PGDH as a key driver of age-related muscle loss, work that has already progressed to phase 1 clinical trials through Imperium Bio, a company co-founded by Dr. Blau. In the November 2025 Science paper, they asked whether the same protein was involved in age-related cartilage loss. The answer was yes, and the results exceeded what even the researchers anticipated. Blocking 1,5-PGDH activity using a small molecule drug injected either into the abdomen or directly into the knee joint caused thinning, aging knee cartilage to thicken again across the entire joint surface in older mice. The regenerated tissue was confirmed to be hyaline articular cartilage, not fibrocartilage, scar tissue with structural features closely resembling the cartilage of young, healthy animals. The Stanford team also tested the approach in injury models that mimic ACL tears in humans, the type of injury that leads to osteoarthritis in approximately 50% of affected adults within 10 to 15 years. In these injury models, repeated injections of the 1,5-PGDH inhibitor twice a week for 4 weeks significantly reduced the likelihood of osteoarthritis developing. The mechanism works through prostaglandin E2, a signaling molecule that plays a nuanced and dual role in cartilage biology. At very low concentrations, prostaglandin E2 promotes cartilage homeostasis and supports the anabolic activity of chondrocytes. At higher concentrations associated with the inflammatory state of osteoarthritis, it contributes to cartilage degradation. 1,5-PGDH is the enzyme that breaks down prostaglandin E2, and its age-related increase in joint tissue progressively depletes the prostaglandin E2 that chondrocytes need for their homeostatic functions. By blocking 1,5-PGDH, the researchers restored prostaglandin E2 to the local concentrations that support chondrocyte anabolism, causing the existing chondrocytes to change their gene expression from a degenerative to a regenerative program and producing the dramatic cartilage rebuilding observed in the study. Critically, the regeneration did not involve stem cells. The chondrocytes already present in the aging cartilage were being reprogrammed into a healthier, more regenerative state rather than replaced by transplanted cells. This is a fundamentally different and potentially more clinically scalable approach to cartilage regeneration than any previous therapeutic strategy. As Dr. Blau stated, this is a new way of regenerating adult tissue, and it has significant clinical promise for treating arthritis due to aging or injury. Before we continue to the stem cell story and the clinical trial landscape, here is a pause for you. Did you know that a study published in the journal Science in November of 2025 described the most complete cartilage regeneration ever documented with a pharmacological intervention? Drop a comment below right now and tell us whether you had heard about this research before watching this video, and whether you are managing osteoarthritis yourself or for someone you love. We read every comment, and your engagement tells us what this community most needs to understand. The stem cell story for cartilage regeneration is older and further advanced than the 1,5-PGDH discovery, and it centers on Cartilage, a product of the South Korean biotechnology company MEDIPOST that combines allogeneic human umbilical cord blood-derived mesenchymal stem cells with a hyaluronic acid hydrogel carrier that has been approved and commercially used in South Korea since 2012 and administered to over 36,000 patients. Cartilage is administered through an arthroscopic surgical procedure. The knee joint is opened, the damaged cartilage is prepared, and the Cartilage product is applied directly to the cartilage defect site, where it adheres and stimulates the differentiation of the stem cells into chondrocytes that produce new cartilage matrix. The mesenchymal stem cells are allogeneic, derived from umbilical cord blood donors, meaning they are available as an off-the-shelf product rather than requiring cell harvesting from the patient. The clinical evidence from South Korean use of Cartilage is substantial. Phase 3 trials conducted in South Korea found that 97% of recipients showed significant improvement in cartilage restoration on MRI and arthroscopic evaluation at 48 weeks, with follow-up data extending to 7 years, showing durable cartilage regeneration and sustained functional improvements. The FDA Phase 3 trial cleared in early 2026 will enroll patients with Kellgren-Lawrence grade 2-3 knee osteoarthritis across more than 60 clinical sites in the United States and Canada. As of June 2026, MEDIPOST secured FDA agreement on a single pivotal Phase 3 study strategy that the company expects will significantly reduce the development timeline and facilitate expedited commercial entry to the United States market. The broader field of disease-modifying osteoarthritis drugs was comprehensively reviewed in the European Journal of Pharmaceutical Sciences in February of 2026, which catalogued all current clinical trials of pro-anabolic intra-articular agents compounds that stimulate cartilage regeneration by inducing the differentiation of mesenchymal progenitors into chondrocytes through a process known as chondrogenesis. The review confirmed that while no intra-articular disease-modifying osteoarthritis drug has yet received regulatory approval anywhere in the world, the current clinical pipeline is more advanced than at any previous point in the history of osteoarthritis treatment. Among the approaches most advanced in the clinical pipeline, beyond CARTISTEM, are the adipose-derived mesenchymal stem cell injections studied in multiple Phase 2 trials. A Phase 2 trial published in Stem Cell Research and Therapy found that intra-articular injection of 100 million allogeneic adipose-derived mesenchymal stromal cells produced significant improvements in clinical outcome measures and significant reductions in inflammatory biomarkers at 1 year, with MRI evidence of cartilage changes consistent with partial regeneration. Platelet-rich plasma injections continue to accumulate evidence in 2025 and 2026, with systematic reviews confirming superiority over hyaluronic acid and corticosteroids at 12-month follow-up for knee osteoarthritis. The question of what adults with osteoarthritis should ask their orthopedist or rheumatologist right now deserves specific and practical guidance. For adults with mild to moderate knee osteoarthritis, Kellgren-Lawrence Grade 1 to 2, the questions most relevant to the current research landscape include: am I a candidate for platelet-rich plasma injections given the current evidence? Is my disease severity in the range that would qualify me for the CARTISTEM Phase 3 trial if it is being conducted at a site near me? And is there any clinical trial of a disease-modifying osteoarthritis drug currently recruiting patients with my grade of disease? For adults with moderate to severe osteoarthritis being advised to consider total knee replacement, the specific question to raise is: has comprehensive non-surgical management been fully optimized before surgical referral? Research has documented that a significant proportion of patients referred for knee replacement who receive optimized non-surgical management reduce their pain and improve their function sufficiently to defer surgery by years or eliminate the need entirely. The honest answer about timeline involves acknowledging a gap between scientific discovery and clinical availability that is longer and more uncertain than the enthusiasm around any individual research finding suggests. The 1,5-PGDH discovery at Stanford is currently a mouse study. The pathway from a compelling mouse result to a safe, effective, FDA-approved human treatment typically takes a minimum of 7 to 10 years, and many promising preclinical findings do not survive the transition to human trials. CARTISTEM is further along with regulatory approval in South Korea and FDA clearance for Phase 3 in the United States, but the earliest plausible timeline for United States approval, based on the current trial timeline with 2-year follow-up as the primary endpoint, would be 2029 to 2030 at the earliest. The mitochondrial health of existing chondrocytes supported by CoQ10 vitamin D and omega-3 fatty acids is directly relevant to their regenerative responsiveness. Chondrocytes with compromised mitochondrial function show reduced responsiveness to anabolic growth signals and reduced capacity to synthesize the matrix components that regeneration requires. The matrix GLA protein carboxylation supported by adequate vitamin K to preventing pathological calcification of articular cartilage preserves the structural quality of the cartilage environment that regenerative therapies will work within. The comprehensive management approach is therefore simultaneously a therapeutic strategy for today and a preservation strategy for tomorrow. Every day of optimized management is a day of reduced cartilage degradation, a day of preserved viable chondrocytes, and a day of maintained structural joint integrity that increases the potential benefit of whatever regenerative therapy eventually becomes available. The 1,5-PGDH finding is particularly relevant in this preservation context because its mechanism acts through the existing chondrocytes, the cells already in the cartilage. This means that the regenerative potential of a 1,5-PGDH inhibitor treatment is dependent on having sufficient viable chondrocytes remaining in the cartilage to respond to the drug's reprogramming signal. Cartilage that has been allowed to progress to end-stage disease with near-complete loss of the cartilage layer may have insufficient cellular substrate for the 1,5-PGDH approach to work effectively, even if the drug reaches clinical availability. Preserving what you have is the best preparation for the treatments that may eventually help you rebuild. The Stanford research team has stated their intention to initiate clinical trials for cartilage regeneration as the next step, building on the Phase 1 safety data already available for 1,5-PGDH inhibition in the muscle weakness context. For adults who want to stay informed about clinical trial enrollment, the National Institutes of Health Clinical Trials Registry at clinicaltrials.gov is the most reliable source of information about trial enrollment status, eligibility criteria, and participating sites. The future of osteoarthritis is in the process of being written by researchers at Stanford, by the engineers at MEDIPOST, by the scientists reviewing every clinical trial in the disease-modifying osteoarthritis drug pipeline. The story is not finished, but the chapters being written in 2025 and 2026 are more promising than anything this field has produced before, and understanding those chapters specifically, scientifically, with clear-eyed assessment of what the evidence shows and what it does not yet show is exactly the kind of health intelligence that this channel exists to provide. The nasal chondrocyte tissue engineering approach represented by the ENCANTO trial launched in 2025 at University Hospital Basel deserves mention as one of the most innovative technical approaches in the cartilage regeneration pipeline. The concept involves harvesting a small biopsy of cartilage cells from the patient's nasal septum, a site where chondrocytes retain a more robust capacity for proliferation and matrix production, and age-compromised joint chondrocytes, expanding those cells in the laboratory, seeding them onto a collagen scaffold, and implanting the resulting engineered cartilage tissue at the site of joint cartilage damage. The nasal chondrocyte approach offers a potential advantage over approaches using joint-derived cartilage cells because nasal cartilage cells retain their chondrogenic capacity more reliably with age than joint chondrocytes. The engineered graft may produce more consistent cartilage matrix quality across the age range of osteoarthritis patients, including older adults in whom joint chondrocyte function is most compromised. The ENCANTO Phase 2 trial will run through 2029 to 2030 and compare nasal chondrocyte engineered cartilage implantation against standard of care for patellofemoral osteoarthritis. The gene therapy dimension of cartilage regeneration represents the frontier of the field. Approaches that use viral or non-viral vectors to deliver therapeutic genes directly into joint cells, programming them to produce the growth factors, matrix proteins, or anti-inflammatory molecules that the osteoarthritic joint environment lacks. The interleukin-1 receptor antagonist gene therapy approach, delivering the gene for the natural IL-1 antagonist that blocks the inflammatory cytokine most directly responsible for cartilage destruction, has been studied in multiple early phase trials, and the growth factor gene therapy approaches delivering genes for transforming growth factor beta, insulin-like growth factor 1, and fibroblast growth factor have shown proof of concept efficacy in animal models, with early phase human studies beginning. The patient advocacy dimension of the 2026 cartilage regeneration landscape raises important questions about how individuals living with osteoarthritis can stay informed about emerging research and future developments in joint health. As scientific understanding continues to evolve, researchers are exploring a variety of experimental approaches, including regenerative medicine, tissue engineering, and novel biological pathways that may contribute to future advances in the field. While many of these approaches remain under investigation and are not yet part of standard clinical care, they reflect the growing scientific interest in improving our understanding of joint function and cartilage health. Ongoing studies will help determine the safety, effectiveness, and appropriate applications of these technologies in the years ahead. For people living with osteoarthritis, staying informed through reliable medical sources and maintaining regular communication with qualified healthcare professionals remains the most appropriate way to navigate current treatment options and follow future developments. The research discussed here represents an evolving area of scientific inquiry rather than established medical recommendations. Understanding where the science stands today and how it may develop in the future can help individuals better appreciate the progress being made in joint health research, while maintaining realistic expectations about the timeline and outcomes of ongoing investigations. Represents a genuine opportunity for appropriately eligible patients to access a therapy with an established safety record and a compelling efficacy track record in the South Korean population. Adults with moderate knee osteoarthritis who have not achieved adequate outcomes from conventional management should ask their orthopedist specifically whether the CARTISTEM trial is enrolling at their institution or at a nearby site, and whether their disease severity and absence of the trial exclusion criteria, which include recent hyaluronic acid, PRP, or stem cell injections make them eligible. The bone marrow aspirate concentrate injections (BMAC) represent another intra-articular biological approach being used in clinical practice at some centers outside of formal trial settings. Based on the principle that bone marrow aspirate contains mesenchymal stem cells and growth factors that may promote cartilage repair. The evidence for BMAC in knee osteoarthritis is less mature than for PRP, with fewer well-controlled trials and more variable results across the studies published, and the appropriate positioning of BMAC in the osteoarthritis treatment hierarchy is still being defined. For adults considering BMAC outside of a clinical trial setting, understanding that this approach has less regulatory validation and less standardized preparation protocols than CARTISTEM or PRP is an important part of the informed consent discussion with the treating physician. The economic dimension of emerging cartilage treatments is one that deserves honest acknowledgement, because the cell-based and gene-based therapies at the frontier of the field are substantially more expensive than the established pharmacological and injection options currently in use. CARTISTEM in South Korea is priced at a level that reflects the manufacturing complexity of a cell therapy product. And United States pricing for a cell therapy product of this type, if it receives FDA approval, would likely be substantially higher. Insurance coverage for novel cell-based osteoarthritis treatments in the United States will depend on the strength of the clinical evidence generated in the Phase 3 trials and the FDA's regulatory determination of the risk-benefit profile. For adults for whom the economic barriers to cell-based therapies may be significant, the scientific relevance of the 1,5-PGDH discovery is particularly meaningful because a small molecule drug that blocks a specific enzyme is, by its nature, manufacturable at far lower cost than a cell therapy requiring the harvest, expansion, quality control, and cryopreservation of biological cells. If the 1,5-PGDH inhibitor successfully progresses through clinical development and receives regulatory approval, its potential for broad accessibility as a molecule rather than a living cell product could make disease-modifying osteoarthritis treatment available to a much wider population than cell therapies are likely to reach in the near term. This is the complete landscape of cartilage injection science in 2026, the Stanford Medicine discovery that may redefine how we understand cartilage aging and regeneration. The CARTISTEM stem cell therapy that is advancing through the most rigorous regulatory pathway in the most competitive pharmaceutical market in the world, and the broader ecosystem of disease-modifying approaches: adipose stem cells, gene therapy, tissue engineering, platelet-rich plasma, that collectively represent the most active and most promising period in the history of osteoarthritis research. The person watching this video who has osteoarthritis is not simply a passive recipient of whatever treatment medicine eventually delivers. They are the stakeholder whose daily management chooses the exercises, the diet, the supplements, the appropriate use of pharmacological support, are determining the condition of the joint that these treatments will eventually work with. The preserved cartilage, the maintained chondrocyte viability, the reduced inflammatory environment, and the structural joint integrity that comprehensive non-surgical management protects are the biological substrate that the regenerative medicine of the next decade will need to succeed. Take care of that substrate. Follow the evidence-based management program. Stay informed about the research, and know that the science is working genuinely rigorously, with results that justify the hope that the 2025 and 2026 findings have generated. Your joints are worth the work, and the research is worth the wait. If this video gave you the honest, evidence-based picture of where cartilage regeneration science actually stands in 2026, please support this channel. Hit the like button right now. Subscribe if you have not already. Share this video with someone managing osteoarthritis who wants the real science about what is happening in research rather than misleading headlines. And leave a comment below. Tell us whether you had heard of 1,5-PGDH CARTISTEM or the intra-articular pipeline before this video, and what questions this research raises for you. We read every comment. Your engagement shapes what this channel creates next. Thank you so much for watching. See you in the next one.